The third part of this presentation is on the Rosetta space mission. We participate with one instrument. On the Rosetta lander, we analyzed cometary organic material, from which we assume that we know the mechanism of formation, and then we can compare the real data from the cometary mission Rosetta with those data that we obtained in the laboratory, simulating the formation of first organic molecules, of first chiral molecules, and these first chiral molecules not in the atmosphere or in the hydrosphere or on planets or somewhere, in the entire universe. So chirality, what is that? Chirality comes from the Greek word which is chir. Chir means hand, and as you know, you have a right hand and you have a left hand. So you take a mirror image of your right hand, and this becomes your left hand. But this does not necessarily mean that your hands are chiral. What makes them chiral is that the left hand at the same moment is not superimposable to the right hand. This makes them chiral. Just imagine you take a sheet of paper. And you ask yourself, is a sheet of paper chiral? So you take a mirror image, you produce a left form of the sheet of paper, but then you recognize that the left form is superimposable, as well superimposable to the original right form. And as a consequence, a sheet of paper is non-chiral. Your hands are chiral. So in chemistry, we have a number of professors that work on chiral phenomena, and this has a name. This is called stereochemistry. I always say one chemist out of 3 is working on stereochemistry phenomena. So in chemistry, this is a very well-known phenomenon, chirality. Many people working on this phenomenon. So for our students, we have a lot of exercises. We have, for example, the question, if your hands are chiral, are your feet chiral? And the answer is yes. Are your ears chiral? The answer, les oreilles, right? The answer is? Not so shy, please. Yes. The answer is yes, your ears are quite chiral. What about your nose? Is your nose chiral? No, the nose is not chiral because you have something which is called a plane of symmetry inside your nose. And you have a lot of symmetry operations that you can perform or you cannot perform with chiral objects. So this is in the microscopic world, in these objects. You can ask the question, are those chairs, for example, chiral? Or this table? Or the video projector? Or the slides? Whatever. These are typical questions in stereochemistry. And our students have to answer them. Then you can move down to the microscopic level, because the same phenomenon also exists for molecules. You have chiral molecules, and the archetype of the chiral molecule is an amino acid which is called alanine. Alanine exists, and the amino acid alanine exists in 2 chiral forms, right alanine and left alanine. If you now try to characterize those 2 molecules, those 2 we call them enantiomers. So you characterize the right enantiomer and the left enantiomer of alanine, and the result is that they have the same color, like your 2 hands. They have the same molecular weight. They have the same, and the same is exactly the same enthalpy of formation. So if you run into a chemistry lab and you try to synthesize the amino acid alanine by a mechanism which is called the Strecker mechanism, students easily can synthesize amino acids, and then you can analyze those amino acids, and you can ask yourself, what did I synthesize? Are these the left amino acids or right amino acids or something in between? And you analyze, and what do you produce is, because both enthalpies of formation from the right and the left amino acids are identical, you produce both amino acids in exactly 50-50 ratio. And this exactly 50-50 ratio you call a racemic mixture, a racemate, exactly. Yeah, all this is a little boring, I know. What makes it interesting now is when biology comes into play. So all this you know. You have enantiomers, you have a racemic mixture if you produce them in the lab. Now biology comes into play. And as you know, alanine, our archetype chiral amino acid, also exists, as Christine mentioned before, in proteins. In proteins you have 20 different amino acids. One of these 20 amino acids is alanine. So you take your protein, you perform a reaction which is called a hydrolysis, which means you cut your protein into individual amino acids. So you have 20 different amino acids. Then you analyze them in order to know, are they right or are they left or are they, what you would expect, racemic? Because always when you try to produce them in the lab, you make them racemic, 50-50. And as a matter of fact, and this is a big surprise, maybe for some of you, not for all, the amino acids occurring proteins are all left amino acids. There is not one single right amino acid in a protein. And the big question is, how did this come? What is the origin of this phenomenon, which is called homochirality? Some people say, oh, this is very easy to answer, this is just random. You take a racemic mixture, you synthesize a protein, the first protein that formed used just by chance left forms of amino acids, and this was the first protein, and then life spread, and this was the origin. There we say this was probably not the case, because if you try to produce a small dipeptide, tripeptide, tetrapeptide, small peptides in a solution of amino acids, and this solution is racemic, you cannot produce long chains. You produce, for example, first amino acid is L, second L, third one is L, you have some L, then you get something which was presented before, which is an alpha helix, and then you, just by chance, you add an amino acid which is of wrong configuration, which is D, and then the chain elongation, the growth of this small peptide stops. The growth is interrupted, and this is what we call enantiomeric cross inhibition. So the wrong enantiomer inhibits the growth of this small peptide to form a protein afterwards. So if you perform the same reaction in a solution of amino acids which is homochiral, which just contains, for example, the L amino acids, or just contains exclusively D amino acids, then you can grow peptides easily. You can form long peptides, which you cannot do in a racemic mixture. This is why we assume the question on the origin of homochirality is strongly associated to the question on the origin of life on Earth. We assume, in order to understand physicochemical processes that are the basics for the origin of life on Earth, we have to understand the origin of homochirality before. When I say homochirality before, let's say we have to understand the first symmetry breaking before. We could produce a small peptide if we have an enantiomeric excess of 80 to 20% these amino acids. You do not necessarily require 100 to 0. So this is why we assume that this question of chirality is strongly associated to all questions on the origin of life on Earth. So our main question at the laboratory, at our lab in Nice, is how can we break chiral symmetry? Yeah, so this we have seen. You have a mirror, you have these two hands, and you have two molecules, and these molecules are chiral. So this is a question that exists in more than 50 years in natural sciences. And as it is illustrated here, in between chemical and biological evolution, you have something called the origin of the chiral bias. This is just what I'm going to explain you. And we have some different models on how this chiral bias can be generated. The first model is on statistic fluctuations, the second one is on the parity violation by the weak force, and the third one is on chiral fields. We work on these chiral fields. So today I would like to present you these chiral fields. We work with what is called light. We use circularly polarized electromagnetic radiation to transfer the chirality of the light towards organic molecules. I will have lots of examples for this later on. Doing so, we can generate a small enantiomeric excess in some percent, and we require amplification mechanisms of this small enantiomeric excess to obtain enantiomeric enhancement. All this is very much related to the origin of organic molecules in space. And this is what makes, in our research, cometary nuclei extremely important. Here you see comet Hale-Bopp, an image from 1984, and you see comet Hale-Bopp with two tails. This is a tail which is called the type 1 tail, the plasma tail, and here you have the type 2 tail, which is a dust tail. We can talk about hours about these two tails, where do they originate, etc. The only thing that I would like to tell you now is that this is in white color, if you like, because this is just a reflection of starlight. What you see here in blue color is a real emission line of carbon monoxide plus cations. The size of this tail here is several hundreds of millions of kilometers. So you require a big source of carbon to generate a plasma tail of this type. The origin of the carbon in the CO2 plus cations in the tail, the origin of this carbon is in the cometary nucleus. So this is one argument for us to say we would like to study the chemistry, the organic chemistry inside the cometary nucleus, because there seems to be a lot of organic carbon. Here's another image of the same cometary nucleus. You can't see the nucleus. The nucleus is extremely small. The nucleus is several kilometers in size. This here is several hundreds of millions of kilometers in size. The type 1 tail, the plasma tail, and here you have the dust tail. Just imagine, I just show you the images due to this blue color of carbon monoxide plus ions composing the blue plasma tail. So, in order to understand now the formation of first carbon-carbon bonds in the universe, we enter into interplanetary space, and we use a model from Meyer-Greenberg from Leiden University. Meyer-Greenberg said, in this interplanetary environments you have small dust particles, they were called IDPs, interplanetary dust particles, here in brown color, and you have some molecules like these molecules, and you can detect them in the gas phase, because in the gas phase those molecules, they do rotations and they do vibrations, and this you can detect by radio astronomy and by infrared astronomy. So here, for example, you have one molecule. Does anybody of you know the name of this molecule here? Any guess, please? CO2, yes, this is CO2. This is not water, because water is not linear, right? So here we have carbon monoxide, water, this one is water, here we have methanol and other molecules. These molecules that I presented here are well known for their presence in interstellar space. We know the environments where they are, we know the regions, we know their density. So this is not rough. This is known because these molecules are in the gas phase and they perform rotations and vibrations. Greenberg said these molecules from the gas phase interstellar space, they will condense on the surface of these IDPs. They will condense on the surface, forming ice layers. And during this condensation, the molecules are irradiated with ultraviolet light, indicated here by these flashes. This light is also known very well. We know the energy, we know the intensity, and we also know the polarization of this light. So we know those molecules, we know the dust particles, the size of this dust particle, I didn't mention that, but the size is smaller than a millimeter, even smaller than a micrometer. So it's a tenth of a micrometer in average size. Later in my presentation you will learn that these particles with their individual ice layers are extremely important to understand the formation of cometary nuclei, because a cometary nucleus is just an agglomeration of these tiny particles with individual ice layers. So a cometary nucleus that is not composed of some mineral inside, some mineral rock with an icy layer outside. A cometary nucleus is an agglomeration of billions of small dust grains with individual ice layers, according to the Meyer-Greenberg model. Okay, so then we assume that there might be some photochemistry going on in these ices at extremely low temperatures. So we simulated these processes in our lab. We have water, carbon monoxide, carbon dioxide, ammonia and methanol. So these molecules you have seen already. These molecules are in the gas phase, and we know their presence in interplanetary and interstellar space. We condense those molecules on the surface in a high vacuum. The surface is cooled down in this experiment to minus 261 degrees Celsius. So this is 12 Kelvin. Colder than Arcachon. And during condensation, those molecules are irradiated with ultraviolet light. This ultraviolet light we know. Here you see the experimental setup in the lab, the high vacuum chamber, the deposition tubes, our lamp that was used. And in this first experiment we used light of energy 10.2 electron volts, which corresponds to 120 nanometer. So this is one line which is known to be the Lyman alpha line. So this Lyman alpha line, of course, is very present also in interstellar environments. We analyzed the ice by using a technique which is called gas chromatography. In gas chromatography, you have the retention time and you see a lot of signals. And this gas chromatography is coupled to mass spectrometry. So, and this allows us, this mass spectrometry, for each signal, for example, to take the mass spectrum, and the mass spectrum tells us the identity of the signal. In this chromatogram, you see a lot of amino acids. For example, alanine, D and L, glycine, aminobutyric acid, valine, beta-alanine, proline, aspartic acid, serine. So here you see 6 amino acids that belong to the 20 amino acids that are parts of proteins. We found a second group of amino acids that we found that was unknown before, that we called ourselves diamino acids, because these amino acids contain 2 amino groups and 1 carboxylic group. This, for example, is 2,3-diamino propanoic acid. Here's another enantiomer. We can separate enantiomers. We see L-alanine and D-alanine, those 2 enantiomers here. For the specialists among you, and next question is this one here. The very first amino acids eluting at a retention time of about 4 minutes has the abbreviation SAR. What's the name of this amino acid? So this one here, ALA. This of course alanine, right? This you know well, alanine. We discussed this already, also Christine before. GLI is called glycine, aminobutyric acid, valine, proline, serine. So this is called sarcosine. Yes, according to our French president. And Athena, don't laugh too much, because afterwards I have some jokes about François Hollande in the presentation as well. So this is sarcosine, and Athena, she recognized sarcosine is the first eluting amino acid, and it's also the smallest one among all of them, right? Before we published these data, we wanted to know, do these amino acids, so you saw 16 amino acids forming in the ice. We wanted to know, do these amino acids form from those individual C1 and N1 units that we deposited, that we condensed, or do these amino acids maybe form from contamination of this chamber? You know, in each laboratory you have some professors moving around and they don't know exactly where they can touch something and where not. So we said before publication, let's do the experiment a second time. And in the second experiment, we replaced the carbon-12 isotopes in carbon monoxide, carbon dioxide, and methanol by carbon-13 isotopes. So we made the same experiment a second time, just replacing 100% carbon-12 by 100% carbon-13. We again found 16 amino acids, and because we use mass spectrometry in the second dimension, we can then say, alanine, for example, does it show the same mass in carbon-12 and carbon-13? This would mean that the alanine is a result of contamination, you know, by some aveugle professor passing. But if it's made of carbon-13, you will see it in the mass signature of the mass spectrometer, and then you can deduce that this alanine was produced from the C1 and the N1 units that were introduced into the system. And here you see the masses from the carbon-12. Here you see the masses of the carbon-13 sample, and you recognize immediately, in the case of glycine, which is one amino acid, you have the molecular mass of 175 in the carbon-12 sample and 177 in the carbon-13 sample. And this means in glycine, which is the most simple amino acid, you have 2 carbon atoms. And this gives you a mass difference of 2. So the produced glycine is 100% the result of these small carbon monoxide, carbon dioxide, methanol, ammonia, and water molecules. So there is no doubt that this variety of amino acids is the result of contamination. So this gave a buzz in the press, also in France. Le Monde, for example, said, des briques du vivant dans un cosmos artificiel. Everywhere it was in the news, and this gave us a lot of funding in our small laboratory in Nice, and we could achieve a new instrument, which is this one. Here you have again gas chromatograph, but this gas chromatograph has 2 dimensions, and this is what I say in analytical chemistry today, a revolution. Maybe some of you know gas chromatography, what you saw before, these amino acids, 40 minutes retention time and all the signals. This is classical gas chromatography. Worldwide today we have running 400,000 gas chromatographs. You can imagine in France maybe 1/50 of them. So if you want to discover something new in gas chromatography, it's not that easy. Except if you do multidimensional gas chromatography. So I say there's a revolution going on today in analytical chemistry, and this is driven by multidimensional gas chromatography. This is one of these instruments. The price of it is 300,000 euro. And the working principle is this one here. You have a first capillary column where analytes pass. They are connected by a modulator to a second capillary column. Have a look at this image here. You have 3 analytes, 3 molecules you would like to separate. And these molecules are called alpha, beta and gamma. As you see on this last line here, alpha is not well separated from beta, which is itself not well separated from gamma in classical monodimensional gas chromatography. If you do multidimensional gas chromatography, you have a second dimension here. So you trace the retention time in the second dimension as a function of retention time in the first dimension. You trace time as a function of time. And you generate a chromatographic space which is now of 2 or 3 dimensions. And doing so, you can well separate the 3 analytes alpha from beta from gamma. This chromatogram here, this is a gas chromatogram. What you see here is a gas chromatogram we recorded in Nice. And you see, I love this image, you recognize, I love it. This is one of the most beautiful scientific images I have ever seen from our lab. So you have here a retention time as a function of time, and all these signals correspond to amino acids. This is our standard of amino acids. And I say this is a revolution because all this theory in gas chromatography, you have to rethink. You have to rethink, for example, what is a Kovacs index, what is a resolution, what is a separation factor. All this has to be redefined. And nobody today knows the answer. How can you calculate the resolution of a 2-dimensional gas chromatograph? Okay. So this is a standard, here again the standard, and now we use this technique to analyze our ice. And using this technique we do not only see 16 amino acids that we saw before, today we see 30 different amino acids in those ices. And here is one part of the chromatogram. This is a gas chromatogram. Again, you trace time as a function of time. I have one part of a big window of big chromatographic space from 55 minutes to 62 minutes, which is here. And you recognize all these signals are amino acids. This one, for example, is called aminoacetylglycine. Here's another one which is called diaminobutyric acid. This one is chiral, so we have 2 enantiomers. Aminoacetylglycine is not chiral, so just one signal. We expanded from 16 to 30 different amino acids that form in the ice. We also, and this was published, no, this was in 2017, we are able to separate sugar molecules by using multidimensional gas chromatography. And as chemists and biochemists among you know very well, sugar molecules appear in a big variety of different constitutional isomers. And we are able to separate all those constitutional isomers by using multidimensional gas chromatography. And this was then published in Science 2016, and there you see, for example, that we did detect the molecule ribose in the ice formed out of molecules that were C1 units and N1 units. From these small molecules we produce not only 30 different amino acids, we also produce aldehydes, which I did not show, and also ribose, such in this case. Ribose is this one. We also found arabinose, xylose, luxose. We found these C4 sugars, we found C3 sugars in form of glyceraldehyde. So this is not only ribose that we have seen in the ice. So ribose is embedded in a wide variety of other sugars as well. Otherwise it would have been unexpected just to see ribose. We found all these sugars as well. And to come a little bit back to chemical evolution, here's part of DNA, here's a protein. There was a long-standing question, who was first in chemical evolution? Was it DNA, the genetic material, or was it proteins? Walter Gilbert published from Harvard University, Nobel Prize winner, he published in Nature in 1986 saying that it was neither DNA nor proteins, it was RNA. The word was already discussed among you, proposing this RNA world in 1986. Here you have the molecule of RNA, which proceeded, as proposed by Gilbert, DNA and proteins. But Walter Gilbert, and that is interesting, also said that RNA, yes, preceded DNA, but RNA was never the first genetic material on Earth. RNA itself was preceded by pre-RNA, pre-genetic material. And you can well imagine that you have many laboratories worldwide that today ask the question, what was the first genetic material on Earth? This is a big question today in modern science. Here I give you two candidates. There are other candidates as well. These candidates are called diamino acid PNA. PNA is peptide nucleic acid, and here we have amino ethyl glycine PNA. PNA again a peptide nucleic acid. En français, APN, l'acide peptidique nucléique. And you see here, peptidic means you have an amino acid which is bound by a peptide bond to a next amino acid, itself bound by a peptide bond to the third amino acid, etc. And the chemists among you, I assume, recognized already that the monomer of this one here is amino ethyl glycine, our amino acid that we detected in the simulated cometary ice. The monomer of this diamino acid, peptide nucleic acid, is a diamino acid, and this is a diamino acid 2,4-diamino butyric acid. This is the one that you have seen on the slide multidimensional gas chromatography of our eyes. So the molecules that we consider crucial in chemical evolution are formed in the artificial cometary ices. To give you a little more biochemistry background on these images, what makes them fascinating is you can use this molecule, we have it in our lab, this molecule here. You can form a duplex of this molecule with RNA. You can form a duplex of PNA with DNA. And if you form a duplex, Watson-Crick base pairing rules are obeyed. So A, T, C and G bind with the complementary nucleotide bases in DNA. So people assume, this makes people assume that PNA might have been a good candidate that preceded RNA and DNA. So these duplex molecules between PNA and RNA, PNA and DNA, we also call hybrid molecules. You can artificially generate a hybrid between PNA and DNA. And you have well understood that DNA and RNA are found today in biological organisms. This is known. PNA, on the other hand, are entirely synthetic. There's not one PNA molecule that was found in whatever kind of organism. No, PNA is purely synthetic. Okay, so you recognized that we are able to simulate those interstellar ices in the lab. We do see 30 amino acids in the ices. We see a lot of aldehydes in the ices. We see sugar molecules such as ribose in those ices. Now we would like to come to this chirality part again, because in order to produce all those molecules we used Lyman alpha photons, electromagnetic radiation. Since 1998 and a publication by Jeremy Bailey, we know that electromagnetic radiation in these environments, for example the Orion molecular cloud number 1, in the star formation region, is at least partly circularly polarized. This means that the electric field vector moves around the translation axis of your light. It moves around and it can run a right helix and it can run a left helix. So we distinguish between right circularly polarized light and left circularly polarized light. So light, in our publications we often say that photons can be chiral, as molecules can be chiral. Photons can be chiral. We talk about chiral photons, and these chiral photons exist in these star formation regions. So the next step is of course evident. We try to generate circularly polarized light experimentally in France in the synchrotron center, which is called Soleil, and we use their chiral photons to understand whether or not the chirality of those photons can be transferred to our molecules. And this is a tough scientific question because the chiral photons do not have any mass. Photons are massless. Our amino acids or the ribose molecules, of course, they have a mass. So is it possible in general to transfer the chirality of massless photons as it exists in interstellar space to organic molecules, to racemic molecules, for example? This is our question. So we enter into this synchrotron. This is one of these beamlines. And this beamline is able to generate, and this is the only beamline worldwide in the south of France at Synchrotron Soleil that is able to generate this light and also able to detect the polarization via the 4 Stokes parameters of this light. And here you can see we can generate linearly vertically polarized light, linearly horizontally polarized light, diagonal polarized light, and this is what is called circularly polarized light, and here again circularly polarized light. So in the synchrotron center you can generate electromagnetic radiation that we require. And the one in France is the only one worldwide. So then we thought, just imagine you have 2 enantiomers of an amino acid, to keep it simple, alanine, which is the most simple, the archetype of a chiral amino acid. 2 enantiomers of alanine, D and L, and they are chiral, as you know. Now you irradiate with, say, right circularly polarized light, not with left, just with right. So what will happen? Right circularly polarized light, and you irradiate D-alanine. Then you can say, the light, the photon, might be absorbed. This absorption will probably depend on its energy, on its wavelengths. And this is what you can determine by absorption spectroscopy. What will happen with a left enantiomer and right circularly polarized light? The same answer. Right circularly polarized light can be absorbed by a left-handed molecule, but it will depend on the energy, on the wavelengths of the light. The absorption by the left molecule or by the right molecule is not necessarily what is interesting for us. What is interesting for me is what we call the differential absorption. We would like to see, is there some region in the electromagnetic spectrum where one of those molecules absorbs more and the other absorbs less. We are interested in the delta A and not in A, the differential absorption. And imagine then that we are above the photolysis energy of the amino acid. You can then generate an enantiomeric excess by using ultraviolet circularly polarized light. So what we require is delta A, and the question again, what is delta A from these chiral photons? Of course, delta A will be function of the wavelengths as well. If A is function of the wavelengths, delta A will be necessarily function of the wavelengths again. So we require the energy dependence, the wavelength dependence of our differential absorption. And this in chemistry, in physical chemistry, in photochemistry is known to be called a spectroscopy, which is called, anybody knows this? CD spectroscopy. This is circular dichroism spectroscopy. In circular dichroism spectroscopy you measure the differential absorption as a function of the wavelengths. So we went to the literature, and this is for the example of alanine. We went to the scientific literature and wanted to know what is CD, circular dichroism spectroscopy of the amino acid alanine in the entire electromagnetic spectrum in 2010, so not too long ago. And the result is this line here. Everybody measured the CD spectrum of alanine in water. And water absorbs below 190 nanometer, so all this region is not accessible. So we, for the first time, measured CD spectroscopy of alanine in the condensed phase. And there you see that you have big transitions occurring between 130 and 190 nanometer. So the optical asymmetry of alanine occurs in this region. It was hidden before 2010, occurs between 130 and 190 nanometer. We did this not only for alanine, of course we did this for all amino acids and discovered more or less the same things for the other amino acids as well. Then we went a step further and said, this differential absorption you know already. But if you want to study the transfer of the chiral photons, of the chirality of the chiral photons towards organic molecules, you also require information on this G. And G is called the anisotropy factor. The anisotropy factor is not only dependent on the differential absorption, the anisotropy factor is also dependent on the absorption. This is the definition of the anisotropy factor to that time. And we then said the anisotropy factor, of course, will also be function of the wavelengths. So what we measure now is anisotropy spectroscopy, and this is new, and nobody did this before. We say we should not talk about an anisotropy factor G in the future. We should recognize that the anisotropy factor is function of the wavelengths, and all this will be called anisotropy spectroscopy. And this is what we do. So we measure in a synchrotron center the differential absorption, we provide the anisotropy, and here you see the first anisotropy spectrum of the amino acid alanine, which I often use as an example. But we do this for other amino acids, for other chiral molecules, for the entire chiral world, if you like. We perform anisotropy spectroscopy. So what you see here is G. This you know, G is our anisotropy factor. G is energy dependent on the wavelengths, yes. But what is the beauty in anisotropy spectroscopy is that you can also detect, you can immediately read in an anisotropy spectrum the enantiomer excess that you can induce in your analyte, in this case in alanine. And here you see you can, for example, irradiate, what is that wavelength here, is 230 nanometer. If you irradiate at this wavelength, you have a value which is 0 here, so you can irradiate for thousands of years, you will never end up with an enantiomer excess. If you irradiate, on the other hand, here at 200 nanometer, for example, or at this minimum or at this maximum there, then you can generate and then you can transfer the chirality of photons towards those molecules. So the transfer of chirality from chiral photons towards racemic organic molecules is energy dependent, and you require anisotropy spectroscopy to understand the energy dependence of this. So we now want then to think about the chirality on the ices that we produce. And this chirality we can determine by gas chromatography. We use capillary columns. In the capillary columns we have stationary phases. These stationary phases are chiral, and here you see the 3 generations of chiral molecules that are embedded in the stationary phases today. This is the first generation, second and third generation of chiral molecules, such as cyclodextrin molecules that are able then to separate enantiomers on gas chromatography. Some of you may say, oh, this is easy to separate enantiomers by gas chromatography. I say, no, this is not easy. Because just imagine you give me all of your shoes. We put them here. We have a little montagne of shoes. You are 50 students. There are 50 right shoes and 50 left shoes. Right? And it would be rather easy, I would say, to construct a robotic device that will be able to separate, for example, black shoes from brown shoes, right? You can detect the color, or a robot may detect the color, and then you separate what is black, what is brown, and this is rather easy to do. But what would you do to design a robotic device that is able to separate the left shoes from the right shoes? And all of your shoes are different, right? So you require a robotic device that can somehow detect the chirality of your shoe. You see, so this robotic device has to be able to see already in 3 dimensions, because if it's in 2 dimensions, like our sheet of paper, it is not chiral. Your shoes, I have 3 dimensions, they are chiral, so the robotic device is not sufficient just to detect the color, right? And this is what I mean. If you want to separate enantiomers by performing gas chromatography, this is not trivial. You have to have a device that is able to distinguish left from right. So here are the 3 generations, and this is the second most beautiful gas chromatogram I have ever seen from our lab. Look at that. You have, and this is a gas chromatogram, you have the baseline, you have 2 signals, they are sharp such as 2 needles. This is like a duplet in NMR spectroscopy, right? You would not have imagined that this is a gas chromatogram. So you have here sharp like needles, all these signals, and you know what that is? Here are the molecules connected. This is 1, 2, 3, 4, 5, 6, 7. Heptane. This is a molecule heptane for chemists, and on carbon number 3 you have a methyl group. So the entire molecule here is called 3-methylheptane. And what makes this molecule 3-methylheptane special is 3-methylheptane is chiral. So you have the right R 3-methylheptane, and you have the left form S 3-methylheptane. 3-methylheptane, this is just a branched hydrocarbon. This molecule is chiral, and here you have the 2 enantiomers. Here you have the same thing for 4-methyloctane, 3-methyloctane, 4-methylnonane, 3-methylnonane, etc. So we can separate all those molecules. I'm not only talking about amino acids or ribose molecules that we can separate. We can even separate 3-methylheptane. The chemists among you, they say, oh wow, this is good. Why is that good? Because 3-methylheptane is just a hydrocarbon. If you want to, if you want to separate the two enantiomers of 3-methylheptane, you have to branch something to it, to link something to it. What you can do with an alcohol, for example, an aldehyde or an amine, you can easily separate those enantiomers. But here in this case, this is impossible. You cannot easily design diastereoisomers that will give you then the separation. If you're interested, I can tell you this evening how we do that. So because of these competences, we were then selected to be participant in the Rosetta mission, because Rosetta, the space mission from the European Space Agency, is going to land on a cometary nucleus. It's the first mission that was equipped with a device, a robotic device, which is a gas chromatogram, able to distinguish between enantiomers. So this is a column, a cyclodextrin column. This is a size, 10 centimeters something inner diameter. This is a detector. And here you have 2 chromatograms that we recorded, one in 2005, the other one in 2013. So the column had been in vacuum for 8 years, and we tested whether the separation factor resolution, these chromatographic conditions did not change. What I want to tell you now is, again for the advanced students, here is our synchrotron light source with an undulator. Electrons circle around. They are forced on a trajectory by an undulator, so they emit something which is called synchrotron light. You have different colors, you have different energies. Synchrotron light. We pass a xenon filter, noble gas filter. This is again synchrotron light. We have a grating, we select energies, we have a polarizer, and here we come in with circularly polarized synchrotron light. This is a light that we can generate, and we have all in our hands to generate the polarization and the energy of this light. And here, this part you know already. Here we have water, ammonia, and carbon-13 labeled methanol. These molecules are volatile. They occur in interstellar space, and we condense them on this surface. We detect in this ice that forms 30 different amino acids. We detect ribose. We detect arabinos, aldehydes, a lot of chiral molecules. And here's just the example for one amino acid, which is our most archetype amino acid, alanine. We use multidimensional gas chromatography to separate both enantiomers and to quantify them. The first enantiomer, the first eluting one here that you see here, is L-alanine. The second one is D-alanine. If we use left circularly polarized light for the irradiation here of our forming ice, we generate an excess of the first enantiomer, which is L-alanine. This is unpolarized light. If we use right circularly polarized light in form of synchrotron light, we generate an excess of the other enantiomer. And this is our model, our hypothesis, how the origin of homochiral structures on Earth was generated. We say chiral photons in interstellar space were responsible to generate a small enantiomeric excess that was later amplified. Here's an example that this generation of the enantiomeric excess, L versus D, is energy dependent. This is what we discussed before. So you cannot always say that left circularly polarized will you give an excess of the left enantiomer. This is wrong. It is energy dependent. This is what we demonstrated here. Here we do this for different amino acids that all show an excess of the L enantiomer using right circular polarized light, also energy dependent. So as a last slide, or nearly last slide, and this chirality part, you have circularly polarized light that is able to generate an enantiomer excess not only in amino acids such as alanine, but also in other molecules. Summary. We had a journalist from Le Monde who was interested in this. He said, you have meteorites, which is wrong. We have comets, right? He said, we have meteorites and you have amino acids. And these things fall on Earth, and also you have circularly polarized light. But this, of course, it must be before, right? He didn't understand exactly what was the experiment. But this, as you know, is not important, because en bref, la vie sur Terre est de gauche. This is more important information for me. Okay, so this is the first part of my presentation. The second will follow immediately. The first part says you have amino acids that we generate, alpha amino acids, 30 different ones. We have diamino acids. We generate aldehydes, we generate ribose in those ices. And we now continue to work on the chirality of those molecules using circularly polarized light in a synchrotron center. Now you may say, what he's telling us. These are simulated ices from the lab. So we would like to see now the Rosetta mission. The Rosetta mission from of the European Space Agency, launched 2004, visited the cometary nucleus, did they find those molecules? So what is this story about the simulated cometary ice in relation to the real cometary ice? So this is what I'm going to tell you now. This is a Rosetta probe. The dimensions are 2 meters to 2 meters to 3 meters. On top of it, you see what is called the payload support module. On the payload support module, you have all the 10 scientific instruments from the Rosetta orbiter. These instruments, Hervé Cotin knows well, Cosima, for example. We have Midas, Miro, Rosima. So these are instruments, they can dust impact instruments. So they have dust particles, they are on some target, they can illuminate them, they can do mass spectrometry with them using Cosima. So they have a lot of information on the Rosetta orbiter. They have cameras, such as Virtis and the OSIRIS cameras, 2 of the OSIRIS cameras. All of this is facing the cometary nucleus. The Rosetta probe uses solar panels of a size of 32 meters, so the entire probe is extremely big. There are no radioactive heating units, RHUs, used on Rosetta. I work on Rosetta since 1997. To that time, we had some people from our team. There was a discussion. What will be the energy source for the Rosetta? Will this be solar panels, as you see here, with the inconvenience to have a very big probe, space probe? Or can we use RHUs, which are radioactive heating units? So one of us, Stefan Ulamek, he went to Moscow. He was sent to Moscow to ask for a price for a plutonium radioactive heating unit. He came back, said the Russians, they want to have 20 million US dollar. European Space Agency said, oh, this is very cheap, let's buy them. You remember? So finally, there was a long discussion then, which were influenced by environmental issues, particularly in Europe. And as a consequence of this discussion is what it was decided to not use radioactive heating units. It was not the price. And now we have these big solar panels. And of course you have spotted the Philae lander, where our instrument, which is called COSAC, is on later to that, which is here. So again, you have the Rosetta space probe, you have Philae lander, and these 3 guys here mount the Philae lander here. Here you have the image. Philae lander with 3 legs, 1, 2, 3. So this is the cruise configuration of Philae lander. Afterwards, Philae lander will be separated and enter into the landing configuration. And you have the payload support module here. The 2 OSIRIS cameras, Virtis camera. And my question to you now is, do you spot the error in this image? What's the error? Something is wrong. The solar panels are to be implemented here and there. The problem is, the payload, all these instruments from the payload support module face the cometary nucleus, which is here. Philae was attached to Rosetta probe in the wrong direction, because Philae suppose that the cometary nucleus is below. So after separation from Philae and Rosetta, the Rosetta probe had to be turned by 180 degrees to follow Philae landing. And this is true, it's not a joke. So what happened then? In 2004, this was launched. The Rosetta probe passed in the first flyby Earth, in the second Mars, then again Earth, a first meteorite called Steins, and then Earth again. And this is the second passage in the asteroid belt, and there you see an asteroid which is called Lutetia. The diameter of this is 120 kilometers. This was not particularly interesting for the Rosetta teams, but all instruments were switched on for test purposes. And you can recognize here that this is a big crater, a small crater, an impact generated, a landslide, which you see there, and on an asteroid having a landslide of these dimensions was interesting for some astrophysicists in this mission. For me, it was more interesting to see this slide of Lutetia. You know, of course, well that Lutetia was the name of Paris. So all these craters here got names from European cities to the time when Paris was called Lutetia. And of course, you will recognize this beautiful crater which is here, right? Nisaea, it's one of the most beautiful craters on Rosetta. You have another one which is a little more ugly, which is more ugly because you have small craters in a big crater, so you have not very nice boundaries, and this of course is called Marsilia, you recognize. Then the Rosetta probe was given in hibernation in 2011. Beginning of 2014 woke up. And here you see the first images of the cometary nucleus taken from Rosetta spacecraft. This was then in July 2014. You have the nucleus of Churyumov-Gerasimenko rotating. If there is a 3D image, I put a small glass on 3D glasses here, so you will recognize. So here you see the nucleus of the comet 67P Churyumov-Gerasimenko, which has a small lobe on the right, big lobe on the left. The north pole of this cometary nucleus is here. The equator is here. Again, you have the small lobe on this side, some intermediate section which is called Harpy you have here, and the big lobe you have on that side. The small lobe is here. There is some Philae which has an altitude of 800 meters, and the intermediate section Harpy here. The Philae is called Hathor. Here again, small lobe with Hathor, Harpy in the intermediate. This section we called Z. This is a big lobe, the small lobe here. This is one of the images that I prefer personally because, as you recognize, on each slide I put my family name, Meierhenrich. On this one I also put my first name, and you see why? Because my first name is already written on the cometary nucleus. Look here, this is a U, this is a W, and with a little bit of imagination you also see the I. I see the I also there. This is another image where you see the gravitational field vectors pointing in different directions, very different directions. This one here, you see some of these rochers seeming to be scotched at this surface, others you have here seeming to be scotched at this surface here. So when these images were received on Earth, we had two parts of Rosetta scientists. One part of Rosetta scientists like Hervé Cotin, they have their instrument on the Rosetta orbiter. They were crying for help. They said, this is the best cometary nucleus you can ever imagine. You see the morphological structures on its surface. This will give us work for at least 10 or 20 years. And then you had another group of Rosetta scientists to which I belong. We had our instrument on the Rosetta lander, and we panicked because it was not known for us. How can you land on a cometary nucleus from which you even do not know whether this gravitational center is in the inside of its form or even on the outside? And this is illustrated by this image here, all these gravitational field vectors. So here, this is an image taken from SIVAC camera from the Philae lander. You see the solar panel, the Rosetta probe, and the small cometary nucleus. Another image, this is a simulation from the Rosetta probe moving around the cometary nucleus of Churyumov-Gerasimenko. Small lobe, big lobe. On this side, UV is written, and here we wanted to land on the small lobe. And now we start with our 3D glasses. Often it takes a little bit of time that your eyes become adapted to this 3D vision. Maybe what you can do, you can move your head a little bit around, and then you see the 3 dimension of all of that. So the small lobe on the right, the big lobe on the left. Again, small lobe and big lobe. When you see all those images, please take in mind that the luminosity of the cometary nucleus had to be increased, because this is a nice image in 3D, because the surface of the cometary nucleus is black. Is black on a black background. So we had to increase the luminosity that you can see something. The black color of the cometary nucleus is like charcoal, charbon en français. So there's a lot of organic carbon inside the cometary nucleus. So this means this is not only carbon in its inorganic form like calcite or some minerals, there's organic carbon inside this cometary nucleus. So you see different morphological structures on these films here. Some of them show you smooth terrain, other terrains are more rough. Here in the middle, in this what is called the harpy section, you can visualize that this section seems to that time to be more active than the other regions of the cometary nucleus. All these cyclic structures that you see here are not crater impacts. There's not one crater impact detected on the cometary nucleus. These forms are called sinkholes. Another mechanism that forms is not a crater from impact. Here again, you recognize the UWE there. We move away from the cometary nucleus, and here you see another beautiful image on cometary activity. In January 2015, there was a first special issue in Science where you see these different morphological structures of the cometary nucleus. Part 1 of the 3D presentation is finished. There will be a second part, I think, later. And cracks were detected 500 meters in length, 3 meters thickness, so our cometary nucleus will break apart. This is one of these sinkholes. I think end of this week there will be a presentation by Matt Taylor from European Space Agency. He will give you more details on all those images. This image I love. Once the Rosetta spacecraft, having all those instruments looking to the cometary nucleus, was turned around to look into the cometary sky. What was expected to be seen is different stars, because on the Rosetta probe you have 2 star trackers, and those star trackers have the job to orient the Rosetta probe in the 3 dimensions in space. When it was turned around, you can recognize here that all these small particles, more than 350 different particles, are the result of jets, active jets from the cometary surface. So the 2 star trackers that were on board of Rosetta, they were confused. They were no more able 4 times to detect the stars. They were confused by all these dust particles in space. So 4 times in the lifetime of Rosetta, the entire probe had to be moved 1,000 kilometers away from the cometary nucleus. The star trackers were switched off. They were switched on again, booted up, reoriented Rosetta, and then we could move backwards to the cometary nucleus. So now, last part of the presentation is landing of Philae. Here you have Philae, lots of 10 also scientific instruments. Our instrument is called COSAC. COSAC is a gas chromatograph coupled to a mass spectrometer, and the first instrument ever sent to space having chirality device. Philae contained 2 harpoons. Here's a scheme of this harpoon. And on 12 November 2014, this was landing day. I was in Cologne at the Lander Control Center LCC. Others, like Matt Taylor, they were in Darmstadt, where the entire Rosetta probe was. You were in Darmstadt, I assume, as well.
I was in Paris.
Oh, in Paris. Ah, yeah, you were Cité des Sciences. Just, I will have an image later from that. So I was in Cologne, where we had the LCC. This is the landing ellipse, so we wanted to land somewhere here. This was the image Philae separated from the Rosetta orbiter, the image taken from the Rosetta orbiter. Another image taken from Philae, you see the Rosetta orbiter. The orbiter with one solar panel, the other you don't see. Philae had a flywheel inside, which rotated to stabilize the landing axis. And here you see that Philae itself rotated during the 7 hours of landing. This was scheduled. We wanted to have 1 rotation each 8 minutes to get stabilized. This is an image. You have the cometary nucleus approaching Philae. This is 1 feet Philae. This is an image in 3D, and you have the 2 feet of Philae and the nucleus from Churyumov approaching. I was always afraid that we pass close by, that we are not going to land on the surface. The cometary nucleus approaching Philae for landing. This is an image, this is a film made afterwards. These are not original images taken by Philae here. But here is a zone where we wanted to land. This is a beautiful image. Big one from Rosetta probe, the small one taken from Philae. We can see this here again. So this is the point where Philae is going to land. In Cologne, champagne was served. In Paris, Cité des Sciences, François Hollande was following. He was taken by his emotions. He said, Rosetta is such a nice space mission, such a mission of this success you can only design and construct and program in an international collaboration with different European countries, and he is completely right saying that. Another thing one could say, Rosetta is a big success not only because it's international, this is evident, it's also a big success because it's a mission where we have been working in between different generations of scientists. Because many of us were already or had been involved before in another European cometary mission, which is called Giotto. And Giotto visited Comet 1P Philae in 1986. And without Giotto and all this science around Giotto, Rosetta would not have been possible. So there are 10 instruments on the lander, 10 instruments on the orbiter. Each of these instruments have individual PIs. Many of these PIs passed away during that time of construction of Rosetta and were replaced by young PIs which took over then. So it's a very beautiful mission, this Rosetta mission. François Hollande, completely right when he was driven by his emotions, but what happened in Cologne, what we saw, was this image. We expected a SIVA image from Jean-Pierre Bibring from EIS in Paris, having a panorama view of the cometary nucleus after landing from Philae's camera, and we saw that. Before, after, the difference, there are the three signs of these legs, but Philae was missing. We had, yes, Philae bounced several times, landed 4 times. We had the data transfer from Philae to Earth is 30 minutes. So we send data back to Philae is then 1 hour in total. So we had a FSS, a first science sequence, which was automated. So instruments were programmed to perform some operations. Our instrument COSAC was programmed to operate 20 minutes after landing. When this was programmed, we did not assume that we were 20 minutes after rebouncing in an altitude of 500 meters in the vacuum above the cometary nucleus. We thought 20 minutes after landing there might be some molecules in the gas phase, and we would like to inhale those molecules by our mass spectrometry. And this is what you see here. So this is our entire instrument, and between the GC, the gas chromatograph, and the mass spectrometer there is this here, which is called a venting pipe. This is a pipe where we would like to get rid of the helium, the carrier gas in these boxes that is used for the gas chromatograph. We would just like to have organic molecules to pass into the mass spectrometer. So this is an exit pipe. So this exit was opened. Here you see the inner diameter is 20 millimeters. This exit was opened 20 minutes after rebouncing in an altitude of 500 meters. Dust grains, volatile molecules, organic compounds from the cometary nucleus entered this pipe. For the first time since 4.6 billion years, these molecules got temperatures of about 300 degrees Celsius. They entered into the gas phase, into the COSAC ion source, where we performed mass spectrometry. So we measured this mass spectrum on 12 November, 20 minutes after landing. This mass spectrum was measured 2 days afterwards, and this mass spectrum was measured some weeks before. So this is, if you like, our blank. You see one mass at 18, which is water, 28, which is carbon monoxide, and 44, which is carbon dioxide. So this is a blank. So astonishing, even 10 years in space, since 2004. In 2014, the instrument still sees gas phase molecules such as water, CO, and CO2. Also afterwards we see this. So this is our blank. These molecules are not necessarily of cometary, or they are not of cometary origin, because this is on 16 October, before landing. But this here is something different. This is 20 minutes after landing. This is our mass spectrum that we saw. So here we have in black the measured mass spectrum, and in orange we have a mass spectrum where we reconstructed a fit to result in the black spectrum. And for this fit we require 16 molecules, and here they are. Another representation of these 16 molecules, they are alcohols, carbonyls, amines, nitriles, amides, and isocyanides. 16 different molecules, which give us reason to assume that the simulation that we perform with the cometary ice in the lab is very close to what is going on real cometary ices. So Philae continued its journey 20 minutes after our experiment. Philae was then expected to be in this region of the cometary nucleus. We were very lucky that Philae, after landing, provided images such as this image from the SIVA camera, 1 feet of Philae. And here you have a surface having an age of 4.6 billion years. So in astrophysics, cometary research a very important image. This is an illustration performed in Paris, Philae lander, and the position of the Philae lander, which is some kind of vertical position instead of a horizontal position. Later, this was published as a cover page in Science, and later of this, Philae was found by the Rosetta orbiter. And as you see here, the position of Philae is more or less as it was expected by the designer before. In this landing position, our instrument was switched on. We obtained 420 mass spectra. One line of this corresponds to one mass spectrum. This was Friday evening. 1 hour later, the batteries of Philae run empty. Here, Philae's lifetime ended. 1 hour before, we obtained 420 mass spectra. The inconvenience is that in these mass spectra, we do not see a lot of organic molecules. This is probably because we were in a vertical position. The drill did not provide us 1 gram of cometary ice, which we wanted to have in our Cossack sample entry. So there are people that say the drill was empty and provided nothing. And this is what we see, the 420 mass spectrum. I belong to a group of people saying nothing does not exist. After landing and rebouncing, and a second and a third time, you have cometary dust, cometary volatiles, cometary organics everywhere on the surface of Philae. So I can well assume that we do not have 1 gram, maybe we have less than 1 gram, and we are still analyzing these 420 mass spectra to detect organic molecules therein. This is the state of our instrument now. For the future, people from European Space Agency said the philosophy behind our instrument is impressive, because we do not only look for organic molecules and their detection. We go a step further, because if we detect organic molecules, we also want to have information on the chirality of this. And this is the reason why ESA, European Space Agency, asked if the Cossack instrument in a modified form will be implemented into the MOMA, the Mars Organic Molecule Analyzer instrument, landing on Mars in 2020. And this is what is going to happen. If you're interested in this, I wrote 2 books, one on the chirality and the amino acids, the second one on comets. Many people participated, many people sponsored. Thanks then, and thank you for your kind attention.